Wow. Thanks to all of you for completely blowing my mind with awesome talks and inspiring discussions. I'm still very new to this game and learning to drink from the fire hose. So I guess my role so far is just asking naive questions. And I want to start with one, and that is, so if you think about a scenario where treatment just failed in a patient, and imagine we could kind of like have a movie where we wind back time and see where that initial clone that caused the resistance, where that mutation happened in the primary tumor, right? And then we could tease out, for example, the position of that initial mutation or how it's moving through the tumor. Could that actually affect the trajectory of the tumor? Could we even use that somehow in the framework of evolution-based therapies? So that's the kind of question that my RegTech team of interdisciplinary scientists at the new Max Planck Center for Physics and Medicine in Erlangen, Germany, do come and visit us, are trying to figure out, or in other words, how can we somehow integrate physics into the game of evolution-based therapies? What I particularly mean with physics is the inherent collective dynamics that growth induces in these active granular matter systems. So active granular matter systems, what do I mean by that? Let me show you. So if this is a population of cells, right, and resistant clones are conveniently colored in here, we would like to figure out what determines the evolutionary trajectories of this individual subclones. However, many cellular populations are not as dispersed as I presented here, but are actually densely compact, ranging from microbial biofilms, of course, solid tumors, all the way to developing tissues and growing embryos. And these are, of course, very different types of populations, but they do share some underlying physical principles. Think about it. If this blue cell here wants to proliferate and grow, right, it has to exert mechanical forces on its neighbors, push them aside, and then they again have to push their neighbors so that we get some form of collective cell dynamics. Right? This is happening throughout the entire population of cells. So even in the absence of active migration, we get population inflation, or in terms of PopGen, range expansions. Overlaying this spatial structure of the population, we often have spatial gradients, such as gradients in nutrients or oxygen, giving cells at the periphery proliferative advantage over their conspecifics in the bulk. So the question that we're asking is, how do these inherent physical principles change the game of evolution, and in particular, drug resistance evolution? So this is all very conceptual. So let's put some meat to it, right? So let's simulate this. Huge shout out to Paul Macklin and for developing the PhysiCell framework here. And bam, here we have a mutation, right? So, and initially this population is inflating, and despite the fitness cost that this red mutant has, it's still growing in size. And I want you to remember that that's going to be important later on, right? But first of all, let's zoom out, give this a bit more space, right? And now two things are happening. If we cut, oops. So if we continue to grow now, we see that a growth layer is developing. So cells in the center stop to proliferate due to depletion of nutrients. Also, due to the fitness cost, the boundaries of this clone are starting to converge. So it's a purifying selection. And we say, oh, yeah, that's going to go extinct at the front pretty soon. Probably somewhere here, right? But now if we zoom into this and continue that, we see that, no, it's not that straightforward. Surprise, surprise. So this is actually extending this very long tips that are incompatible with how we thought selection is working in these range expansion systems. Okay, so this is, I really love agent-based models, but obviously also you have to do a lot of assumptions. So what I also like to do is test this in experiments. So can we do this in tumor is probably a bit tricky, but we can do it in a different type of dense population. And that's what we do. We like microbial model systems. So such as this yeast colony, right? And I do get it, like a yeast colony is not as complex, Andre, as a real tumor, right? But we're interested in these underlying shared physical principles that all dense populations have in common. The nice thing about yeast is that, well, it's growing much faster, right? While you have one cell division in a cancer cell line, you have 65,000 in yeast, right? Also, it's very genetically accessible so that we can easily, even I as a physicist, can build in genetic constructs. So here the resistant clones are colored in red, and we can tune the fitness, etc. So let's see if this actually also is happening in the experiment. Again, those two boundaries are converging, right? And lo and behold, yeah, so we inherently see these elongated tips. So something is weird with selection here. And if you quantify this, right, you see that we have some type of width-dependent selection, or I think the way that we've been discussing this a bit in the last 2 days is, oh, is this something like local frequency-dependent selection, right? As it turns out, you can understand this by looking at this collective dynamics. And in a nutshell, what's happening is that the collective dynamics, so that cells that are close together have to move in a similar manner, suppresses the differential displacement of cells that selection needs to act, right? Okay, so that's all nice, but we want to put it into mathematical models. And actually here it's nice that you can augment just your standard range expansion or surface growth models with an effective line tension, and you can come up with the same kind of dynamics. Okay, well, you might add that maybe, typically drug-resistant mutants don't come in at these huge chunks, but individual mutations. So we can also do that and disperse resistant cells into here a dark wild-type background. And you again see this very long streaks. And what Sari, a super talented PhD student of mine, realizes, something fishy is going on here. Because if you quantify the width of these streaks, right, you would expect them to grow linearly in size if they're neutral, right? And they do in the beginning, but then they level off. And they stay at a constant size. Although the population is inflating, they're selecting all the jets, right? So what's going on here? In order to understand that, we have to think about which forces are acting on these clones. There's one, inflation. We saw that in the beginning. So trying to drive the boundaries apart, right? And then we have selection on top of this. I just told you that in these dense, collectively growing and expanding populations, selection is not constant, but width dependent. So if we think about this in how the width changes while the radius is expanded as a function of width, we see that if we add the two effects together, we do get a stable equilibrium, where smaller clones are inflation dominated and grow, while bigger clones are selection dominated and shrink. And that explains why we have this constant width. Now that has massive effects for all kinds of downstream stuff. For example, these slower growing resistant cells could, in theory, be rescued by compensatory mutations. But how do we do that in experiment? We do not see mutations unless we build them ourselves. So that's what we did. So what you see here is what we call synthetic mutation. Here you see a slow growing red cell switching to a faster growing blue phenotype, and that's coupled to a fluorescence change, and we can tune the rate and the effect of this mutation. Now we can do funny experiments such as this one, where we retrack the entire evolutionary rescue dynamics of individual clones in these systems. Long story short, if you quantify this, you see that physics increases the probability of rescue and then eventual treatment failure by 2 orders of magnitude. Okay, so of course, then we can put this back into agent-based simulations to see if that still holds true. I'm not going to go into much detail here, but this is what it looks like. And essentially, we recover the same dynamics in these agent-based simulations. And I invite you to check out Sari's paper that we're very proud of. But I now want to switch gears a bit and tell you what we're doing right now. So with the synthetic mutations, now we can look at how heterogeneity arises in these populations, right? By switching from just mixed inoculi to actually growing from an individual cell and then look at these incoming mutations. But we also want to look at therapy dynamics. And that was what Nico is doing in my lab. So he's, this is what you see here. Actually, let's zoom in a bit. So you see incoming yellow mutants all over the place, and we're zooming out, right? And then we initiate treatment and can see in real time how treatment is failing here and where these mutants are coming in and starting to correlate the distance from the edge here with the probability of escape of such an internal clone. Right. We're also pushing this towards 3D now. That's what it looks like. So here, same thing. Now this is a spheroid in a gel. You see incoming resistant mutants, and I like this one in the middle, which kind of was internal and now is pushing out. Well, as much as we like our microbial systems, if we do want to think of cancer more and more, which we are, right? We do have to switch this to one other dimension, and that is the cell type. So what we're doing right now is implementing these types of assays in melanoma tumoroids. So that's what you see here. So this is a gel-embedded, hydrogel-embedded melanoma tumoroid that's undergoing treatment. You can see how it's actually meeting its demise. And as of last week, also, we finished CRISPRing in our BRF-MEK inhibitor mutations and coupling them to colors so that we have now mixed spheroids. Unfortunately, I cannot yet show you nice time lapses. That's still under the microscope as we speak. But in principle, that's kind of what we're looking for. This is a proof of concept experience. So where we have a resistant mutant, then we kill destroy the tumoroid, and then see at the resurgent growth of the resistant clone. Okay, well, you know what's next, of course, right? So can we use this to learn something about adaptive therapy, right? And I just want to briefly switch to something else because we've seen this in illustrations. And Sandy was so kind to volunteer to do this. So this is an interactive real-time simulation, otherwise known as a computer game. So here we have a model tumor comprised of normal susceptible cells and resistant mutants. And when we start this now, right, Sandy can apply treatment, and maybe you can do like maximum tolerated dose first. Just go press it. Yeah. And then, okay, actually, so you just, I'm sorry. So maybe in the interest of time, let's go to, you all know what's going to happen, right? So coming back. So let's do maximum adaptive therapy. And of course, we kind of know the concept, but the question that we're asking now is, should we think about more about a confining shell that we want to maintain in these systems, right? And that sometimes happens. There's a lot of stochasticity in this here, so we can actually extend that. But as you see here, okay, it's breaking out. It's not breaking, although it has a fitness deficit, right? But it's because it's pushing out. And I think what I'm most excited about, thanks a lot. So my students actually can cure the tumor. You can push into the necrotic core and then you actually get a cure. But we get another chance on Friday. So just to finish this up now, one thing that I'm particularly excited about is obviously if we don't have always Sandy's amazing neural net around, right? Is if we can train an artificial neural net to do this. And actually, let me wrap this up and go to the 3D right away. So this is an agent-based model that we trained on mathematical models first, and now we're deploying this towards a 3D agent-based simulation. And I think what I want to show you here is that while we don't see what's happening in the numbers, we see already in the flow. So we have this programmed flow of yellow resistant cells towards the periphery. And we already know that at some point, they're going to escape. They're going to be pushed out of the tumor mass and then start to proliferate. And the question is, can we somehow use these concepts of space and competition and collective dynamics to come up and parameterize them into the mathematical models that then allow us to do better machine learning and come up with better adaptive therapy strategies. It's an exciting journey, and if you also think that's interesting, I hope this can be the start of a conversation. And I thank you very much for your attention, and yeah, for questions now or during the coffee break. Thank you very much.
very fascinating stuff. I have two questions. One, what happens if you put an obstacle, physical obstacle in front of the, to like simulate the scenario of invasion? And second thing, like, to my understanding, in your case, the cells are just pushing if they're inside and they can proliferate if they're when they're outside. But how it would be impacted if there is still some turnover on the inside? Your neighbor is dead and you can like take over the space. Mm-hmm. How would it impact this this interesting dynamics that you're observing?
Yeah, two excellent questions. So first of all, roughness of environment, I think, matters a lot. So we tried this in microbial experiments where you actually have a physical roughness of the surface and you just increase genetic drift a lot. So any bending around an obstacle or squeezing through is a bottleneck. It's a spatial bottleneck. And then after that, you typically have a re-expansion. So because then suddenly, oh, you do have space. And then this clone that whatever that lineage that went through can rapidly expand. And then you have all these inflation effects again, which protects you against selection. So you would expect increased diversity after that. We did some microfluidic experiments where we actually looked at this, but I don't think that's conclusive yet. There are too many ideas, too little time, right? But excellent question. And the second question is turnover. So in these Asian-based simulations, we actually do have turnover, right? So that releases pressure. So it's a combination of proliferation, which shrinks due to the gradients of nutrients, and the release of that pressure by expanding the population. And if you have more turnover, you will decrease that pressure, and that will also decrease the proliferative effects. So one of the things that we're thinking about is, okay, could we have maybe also a spatial gradient of treatment so that we can decrease this collective effects internally, but keep this confining shell around? But I don't know yet how I would do that in a tumor, right? So locally treat the core rather than.
So how would like higher turnover impact the sort of the kind of the balance?
Yeah.
How would it shift the balance?
So how fitness decreases with width, right? Mm-hmm. So the amount of collective dynamics that you have is basically proportional to the width of this growth layer. So how much biomass you generate per surface area, right? And if you have more turnover, then you can actually decrease that if proliferation stays constant. So you get a smaller effective growth layer per se, mm-hmm. right? This is all very ad hoc, so we're really not parameterizing in all the biological complexity yet, but we would like to do that. And also maybe, you can use that and parameterize things into HAL, which is much more efficient in taking care of the biological complexities. So I think that's an interesting trajectory. Yeah.
We should take the question at break.